Thank you. So it's a great pleasure to be here. My name's Stuart MacGowan. I am from the University of Dundee. And today I'll tell you about work that I've done in Jeff Barton's group to combine protein evolution with population genetics. So I'll start by giving you a quick introduction to the field, and then I will show how multiple sequence alignments can be used to essentially increase the power that current human population variant data sets have. And then I'll show how that analysis can be used to detect structural elements in a way akin to classical conservation analysis. But the key point is that we get more from the variant data than we would from conservation on its own. So just before I start, this is Dundee, the city viewed from the law. It's a very nice place. You come in over the river if you travel by train, and on the right, that's the Discovery Centre before we got the Life Sciences sign, which is part of Life Sciences at Dundee, and computational biology lives in there. It's a fantastic place to work with lots of people to potentially collaborate with and explain biology to us sometimes. So diving in, you probably know that there's been a lot of large-scale human sequencing studies done over the past decades. We now have tens of millions of identified genetic variants that exist in a population. And thanks to resources like dbSNP and Ensembl, it's been integrated with genomic annotation, and we can retrieve variants for any region in the genome at will. So a key motivation for a lot of those studies, if not most of them, is disease gene identification, identify variation that causes diseases. But unfortunately, sequencing lots of people is necessary, but not sufficient to do that. So from the protein structure perspective, this is just one example of lots of work that's been done over the years where for protein codon variants, the structure provides a context to understand how the mutation has its effect. So for instance, disease mutations tend to be enriched in the protein core. They can destabilize the overall stability of the protein. Or in this example, they were shown to be enriched at interaction surfaces. Another recent method, which a few groups, wow, that's quite low res. Anyway, another recent example which some research groups have been taken has been to look at spatial clustering of variants and protein structures. And in this particular one, they compared neutral variation, disease variation, and somatic variation, and found that they had different clustering propensities. So neutral variation tends to be over-dispersed than random. Disease variation tends to be clustered. These are very interesting features that make me think that if we can use structural features to, interpret a missense variant or even predict its effect, we could probably go the other way around and use variant densities to predict structural features if we had enough data. Now, on the genetic side of it, they take their own approach. So this is one paper from the Goldstein lab where they developed a method called RVIS. And in this plot, each point is a gene, and they are regressing the amount of common non-synonymous variation against the total amount of variation. So that includes rare variants and synonymous variants, silent mutations, for all genes in the genome. And it's said that genes that have less than the expected amount of common variation that would be proposed to be intolerant to functional variation. So this is because if a variant is common in human, it probably doesn't cause a bad phenotype or it's bad news. So this kind of shows that variant densities work on a gene-gene level. Later, they reduced their unit of comparison to domains, and they discussed the possibility of moving to single residues, but their conclusion was there is not enough data yet, it's too sparse. So the first thing I'll tell you is how we can actually fix that. So achieving protein resolution of the variant distribution. And this relies on the fact that proteins are organized into domains, and domains are related by homology, and they can be expressed in multiple sequence alignments. So in this is a cartoon representation of a sequence alignment, and at the top are human paralogs, and the rest is all the other species in the domain family. The dots represent variants that are found in the human population. Now, if you look at a single sequence, you'll notice that for most positions, there are no variants. It's empty. You couldn't really look at relative densities across a single sequence. However, you add it up over the columns, and you get a lot more power. It depends on the size of the family, but you might have enough power to see meaningful variation in the residue distribution of variants. Thanks to a lot of effort by people building resources and other people in the group helping me to interact with the Ensemble API, for instance, we can do this relatively straightforwardly at this point. So you can connect uniprot residues to variants with Ensemble. You can use BioPython to map the alignment columns to those uniprot residues, and we can pull in other data like conservation scores from resources developed at our group, such as Jabos. That will come in later. So the data sets that I use are PFAM for multiple sequence alignments. So PFAM is a fantastic resource that has over 16,000 protein domain families built by hidden Markov models. But we can, of course, only investigate families that have at least one human sequence, since we're only exclusively using human variation. And moreover, to get any kind of benefit from the aggregation, we need to have more than one. So there's around 6,000 families with at least one sequence, and 3,000, for example, that have more than 5. And we pull in the variation data from Ensemble. Ensemble aggregates variant data from dbSNP, from EXAC, from 1,000 genomes, so it's a one-stop shop for a lot of variant data, and really importantly, functional annotation, so you will have the predicted effect, whether it's a missense variant that changes the amino acid sequence, or it's synonymous, or stopped gained, or frame shift, or we need that information because those different variants will behave differently. And they also have other annotations like, is the variant believed to be involved in a disease, which we can use for testing after. So if we go through that process and map the 6,000, well, first, sorry, this is a before and after slide. So if you look at the variant density on single protein sequences in PFAM, the vast majority have no variants per residue. After the aggregation and looking at columns, most columns in PFAM have 2 to 7 variants, but it depends on how many human paralogs are in the family, and it also depends per column on the occupancy of the column. Some columns have a lot of gaps, and you can't find a variant for a residue which doesn't exist. But I hope you'll agree that this is certainly an improvement and increases our ability to detect useful things. So the first question we had was, do conserved residues have fewer variants? If this method is useful at all, then it should behave in a way akin to evolutionary conservation. So we could check that quite simply, and the answer is yes. So this plot shows missense variants plotted against the Schenken divergence score. So increase in divergence means more residue diversity. And we would expect that in unconserved columns, you would have more missense variants. Now, in contrast, synonymous variants, which are silent at the protein level, act as a negative control. So this is very nice. This is a single protein family in PFAM, But we can do this for all protein families and PFAMs, and these are histograms of the regression components, coefficients, sorry, where for missense on the top tends to be positive, and for synonymous it's typically 0. So we think this works. You might then think, so, okay, I kind of answered this question because what constraints influence missense variation? Well, the same constraints that influence evolutionary conservation, clearly, otherwise there would be no correlation. But for instance, if we look at SH2 protein domains, the red surfaces here are read patches which are depleted in missense variants, and these, in this case, are interdomain interactions. So interactions are depleted in missense variants, and we also see, for example, that secondary structure elements. So on the top, we see in the blue lines are conserved hydrophobics, which line up with the secondary structure elements. These are typical patterns, and we see the same kind of patterns in the missense depletion at the bottom, where the red columns are showing that. Okay, so that's cool and interesting, but the big question is, do we have anything new from doing this, or do we recapitulate evolution? Now, to do that, I'll introduce the concept that I call the conservation plane, which is very generically, you pick a score to represent the missense distribution over the alignment columns, and on the x-axis, you put a score which represents the evolutionary conservation of a column. So again, we have that kind of positive correlation overall. And if you think about this in detail, the concordant areas we could call conserved and missense depleted. This is what you would expect usually for a conserved site. Unconserved, they might be missense enriched, have more missense variants. It doesn't matter what it is. But the diagonals could be interesting if they're not noise, and I don't think they are. So you can have conserved sites which have an excessive missense variation, and probably easier to interpret at the moment are sites that are unconserved but missense depleted. So this means that in that column in the domain there are a variety of different residues there, but whatever is there is constrained to be that in the human population, and that sounds like a functional determinant or a specificity determinant. So we wanted to test that in some way. So now I'll abbreviate unconserved missense enriched UME and unconserved missense depleted UMD. And we can look at the types of interactions. We can compare how frequently you observe certain interaction proteins at these sites. And typically we find that interdomain interactions are enriched in the UMD columns. Ligand interactions are enriched in the UMD columns too. But you have to remember this is lots of different families. If we find the absence of a ligand, it could be that that domain doesn't bind any ligands. So if you do a test where you just generically say, is it involved in an interaction, you get an even nicer result. So that's doesn't prove their specificity, but at the very least it tells you this is a way to stratify unconserved residues. Another very powerful argument, so this is a kind of schematic idea of that conservation plane, and you can look at the relative distribution of pathogenic variants, so annotated with ClinVar, and it would be known, for instance, that conserved residues on the left of the plot are typically enriched compared to other residues. But if you look closely, if it's conserved and depleted, it's even more enriched. And then it would be thought that most unconserved residues are depleted in pathogenic variants, but the unconserved and missense depleted set have quite a high enrichment. There's a high error there because there's few of them, they're quite rare, but it's statistically significant for what that's worth. Okay, so I'll show you a couple of examples of particularly interesting families that have these unconserved depleted residues as we classify them. So the first one is the nuclear receptor ligand binding domain. And this is a ligand activated transcription factor, so there's ligand binding domain and a DNA binding domain. And in this part, we have 4 of the residues. They're marked in blue in the blowout, and we've picked out the ligand binding site. You know, it's an example of that. There's the same ligand binding site in other members in the family, but it's a bit different. But what's interesting, of the 4 that are flagged as this class of variant, 3 of them directly interact with the ligand, and a 4th is on the helix, which interacts with the ligand predominantly, but it points away, it's distal to the binding pocket. You know, I've looked at this closely for a while. So this is it in 4 different proteins. So it's the blue residue as the unconserved missense depleted, and you can see it adopts a variety of different residues are there. But the interesting thing was that there was a strong helical interaction being mediated, which in one member even shifts entirely. And I haven't seen mutagenesis of this position, but we're thinking since this binding domain transduces the signal of ligand binding, maybe this affects how that works. It would be great if someone would mutate it. And then another great family, as an example, are the GPCRs. It's a very big family. This is the family with the thresholds that we use. We found 11 of these sites marked in blue. And you'll notice 5 of them are in a row, and we pick out that ligand binding site straight away. Also, we pick up one of the sodium binding residues, which we're told is very important for class A signaling. And I won't play the second video. There's more that we could say about this. We look at these residues and we see that, for instance, there's a pathogenic site at one of these sites which has been functionally assayed and has been said to be a specific specificity determinant functionally. There's a lot of information on variants besides what I've done here. But I'll finish up and I'll say that, so I've resolved the human population density variant decision to residue level resolution, alignment columns, but you know, you can infer down the way. And it's largely congruent with evolutionary conservation, which is a good sanity check, but it probably shows us something interesting. And we have a preprint available. And I'll finish with acknowledgements. So the group, Jeff Barton for asking hard questions a lot, and the guys that helped with coding and looking at examples, JalView team, and everyone else, and of course resources and people who produce the data. And thank you.
Okay. We are really late, so I will try to avoid the questions. If you have questions, please ask them afterwards. And I give word to Rafi for concluding remarks. And I think we